Last time I was talking about applications of genetic engineering. And we've talked about applications in bacteria, so I'm not going to go over that again. And also, last time I talked about applications in fungi, and I talked about applications in plants, and I talked about applications in animals, and told you a little bit about knockout mice, and I told you about how one engineers goats and sheep and pigs, and then wrapped it up with a discussion of plants. And one of the most important things that I hope I conveyed to you is in talking about any of these issues, whether it's genetic engineering, Max, in bacteria, whether it's genetic engineering in humans, whether it's genetic engineering in animals, one must take a scientific approach in dealing with these things from an ethical or regulatory or whatever. In other words, if you know about the properties of genes and how they work, you should be able to discuss intelligently, at least by now, why these things should be done or why they should not be done. So let me talk about, for the beginning of this lecture, kind of the pinnacle of genetic engineering, and that would be genetic engineering of us. You know, what kind of genetic engineering has been done with humans, what can be done, what will be done, and, you know, clearly this is where the rubber hits the road, so to speak. This is where genetic engineering can have the most impact on humanity, not simply from the point of view of correcting genes, but also from the point of view of treatments, cancer, treatments, viral diseases, and other kinds of diseases, as you will see. And so there's a lot of different aspects to gene engineering in human beings. So let me start out by just defining things in the context of things that you actually know. Because in many respects, there's not any difference, as you know, in engineering a pig or a mouse compared to a human being. The organisms, as we've discussed, develop very similarly. The eggs are organized similarly. The somatic cells are very much the same. And the way you insert genes is very, very, very much the same as well. And so you know from our discussions of genetic engineering in animals that one can inject genes into eggs. You can inject genes right into the eggs of mice. You can inject genes into the eggs of pigs and goats and sheep. And that would be called, in the human context, germline gene therapy. In other words, you'd be adding a gene to the germline, which is the line in all of us that contribute to the production of sperm and egg. And when we do this in animals, Faria, it's permanent. That is, those genes are passed on from generation to generation to generation. And when one is engineering an animal, that's exactly what you'd want. You want to make a transgenic animal. But when you're engineering a human being, that's exactly what you don't want because of a lot of reasons. Ethical reasons of putting a new gene into a human population that you could never get out ever again. And who knows what would happen to that gene in certain environmental contexts. A gene might behave in ways in which are not predictable on the basis of just in one individual in a confined environment. Secondly, as you know from our discussions of transgenic animals and the discussion of cloning and all the things that we talked about in both discussion and lecture, Derek, you know that this is a very inefficient process. And you know that inserting pieces of DNA into genomes is not very precise. And you know that those genes can go to any location of a genome, and if they do, they can cause mutations. And so one has the potential then for inserting a gene in the germline, that is in the egg, of a human being and mutating a gene that might be very important for the development of the human being, and therefore the human being might be very abnormal that you would get out of such a procedure. And so one wouldn't want to do that. And so the point is that you can't predict what's going to happen. The point is that the process is very inefficient. The point is that one can get something worse out in terms of a human being that would have to live their entire life than what you're trying to correct and put in. And then, you know, there's also consent issues. You know, does a human being have a choice of being genetic engineered or not? You're sticking a gene in an egg, and that person's going to be grown up as a genetically engineered human being. They never had a choice in any of that. And what are the rights of that egg? Gets all kinds of murky issues. But I think from a scientific point of view, those of us who consider ourselves genetic engineers would consider this, at this point in history, to be highly unethical and to be something that no one would do for all of the reasons that I've discussed and many, 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 many more. And besides, there may not be any reason to do anything like this. In other words, change the germline because of all these unpredictable things. And it might be, in fact, better to engineer humans by using somatic cells. And so that a person might be engineered, let's say, to correct a disease, but that gene goes away when they go away. It doesn't affect populations. It's not passed on. It's not transmitted into different people, different individuals. It's not perpetuated for all of forever. And therefore, there's a lot of reasons not to do that. And so the second kind of gene therapy, genetic engineering in human beings and in animals, but mainly in human beings. Animals is mainly germline, because we want to make transgenic animals. In humans, it's non-germline, or what's called somatic cell gene therapy, and these are cells like skin cells and liver cells and brain cells and blood cells and all the cells that don't give rise to either sperm and or eggs, non-germline. And there's a variety of things that one can do with this, and I'll discuss a few of them. Some of the things we've alluded to here, but let's just say what we would call gene augmentation or supplementation. You're just adding the correct gene to a cell type to correct for a mutant gene to enhance that particular function. One can also imagine that one could replace, that is, if one can get very efficient with this process, you might be able to replace a defective gene and add a good gene in its place. So that would be gene replacement. Another thing might be you would want to, let's say, shut down a gene. For example, in your discussion tomorrow, you'll see that cancer genes are genes that are on in the wrong time in the wrong place. One would really like to be able to shut them down. And I'll show you how that can be done using somatic cell gene therapy as a potential tool to shut down genes that you don't want on. Or let's say viral genes from a viral infection like HIV or something like that. And so that would be called, in the terms of this slide, target inhibition of gene expression. And I'll also show you how you could use gene therapy to kill specific cells. In any case, there's many different permutations and combinations of somatic cell gene therapy, but keep in mind the difference. Again, germlines, the gene goes in and it never goes away. That's how we make transgenic animals. That's how we make transgenic mice, transgenic goat, transgenic sheep, horse, pigs, you name it. That's how we make essentially transgenic plants. We modify the plant so we get a transgenic plant that perpetuates the gene and the phenotype for generation after generation. With humans, that's a no-no. We don't do that. It's unethical. It's not permitted under a variety of different guidelines. In order to work on humans, and I'll talk about this, you have to get informed consent from the patient. You have to follow your institutional review board. That's called an IRB. And you have to have your protocols signed off by the institutional review board. You obviously have to have funding, and I'll talk hopefully a little bit about that today. And unless you have informed consent from your patient and signing off from your institutional review board, you're not going to get funding from NIH or any other agency because they require these forms as the part of the process of submitting for funding to do these experiments. And you have to have a protocol which has been approved by the recombinant DNA guidelines. And that's where really the DNA guidelines for recombinant DNA are really used, mainly for human somatic cell gene therapies. All of these protocols have to be approved by a recombinant DNA advisory committee under the auspices of the National Institutes of Health. So there's a variety of different issues. Those issues would never fly with a germline gene therapy protocol, but in many respects they fly with somatic cell gene therapy. There's two kinds of somatic cell gene therapy, and it's kind of a weird distinction. Remember, we're talking about two kinds of somatic cell gene therapy, and it's a weird distinction. One's called ex vivo, and one's called in vivo. Of course, they're all in vivo, because they deal with cells, and they deal with living cells, but there's a distinction here. In vivo somatic cell gene therapy, Tom, deals with taking a gene, taking a piece of DNA, and treating, let's say, the patient directly with it. Okay? That would be considered in vivo gene therapy. You're just giving them the gene, and the gene is doing what it's supposed to do. Ex vivo means that you take some cells out of the individual, and you genetically engineer the cells that have been taken out of the individual, Max, and then you put those cells back in the individual. So ex means outside of the body, in means inside of the body. In vivo somatic cell gene therapy, ex vivo somatic cell gene therapy. And ex vivo somatic cell gene therapy would sort of be analogous to what you discussed in discussion last week with respect to inserting a gene in a cell which potentially can become a stem cell, or it's sort of like when we discuss transgenic pigs, engineering the somatic cells of the pig and then cloning those cells. It's kind of like that, but there's obviously no cloning involved, and you're not generating a clone in this particular case. So we have, again, in vivo, taking the gene in any way that you can deliver it. It's usually viruses, as you will see, and just treating the individual directly, or the target tissue, or the target organ, or whatever you want. And then there's ex vivo, where you get the cells out, treat the cells, and then put them back in with the gene that's been added. This is just another version of the same thing. What you can see is that we have this in vivo. The patient is lying in the bed, and you're adding the gene, let's say, intravenously, but you do it in a variety of different ways. And here is the ex vivo. The cells have been taken out. They can be any kinds of cells. Generally, they're blood cells, generally white blood cells, because you can work on them very nicely by bone marrow transplants and things like that. The gene is inserted by transformation or by a virus, as you'll see, transfection. It's put back in, and the cells are put back in to the patient. And here are many of the vectors that are used, and we'll discuss these in again. And most of them are viruses, and the most common form is a retrovirus, which we're going to talk about, and an adenovirus. And adenoviruses are things that cause colds and things like that. So let me give you some in vivo gene therapy examples. And these examples I'm going to give you are things that have been tried and do work and have undergone clinical trials, and in some respects are still ongoing, but people are still doing these kinds of things. So let's talk about cystic fibrosis. Recessive Mendelian trait in humans, devastating. Average lifespan of a cystic fibrosis individual is age 30. I mean, this is really a lethal disease. It is really serious. And that's why one is screened for cystic fibrosis, and Michelle Evans talked about that last week. I've talked about it. If you go back and look in your notes, you'll see how we used ASO, allele-specific oligonucleotides, for screening for cystic fibrosis using PGS, but clearly it's extremely important. It's a very high frequency, as you'll see in Europe, in populations of European ancestry. Almost 1 in 20 are carriers. So anyone of European ancestry in this room, including myself and others, have a 1 in 20 chance of being a carrier for cystic fibrosis, and that means the chance of two people coming together and having cystic fibrosis child is very high, and that means that child has an average lifespan of only 30. That is not very much. And so one would like to perhaps avoid it as much as possible. It's on chromosome 7. As you know, there are these bands on these chromosomes. You've talked about them in discussion, and these bands mark regions of the chromosome. They're markers, remember, markers for chromosomes. And so the cystic fibrosis gene is right here between well, it's within this red mark that I put up here, and here is the gene. It's pretty big. It's 200,000 bases in length. It has many introns that are very large, and you can see mostly the exons are relatively small, 24 exons contributing to the production of the messenger RNA. Here is the protein. This is the cystic fibrosis transmembrane protein. It's a very interesting protein because it is in the membrane of your cells. All of us have this particular protein stuck in the membrane. And why is it stuck in the membrane? It's stuck in the membrane. Tom, remember we talked about how things get in and out of the nucleus? Remember I asked you and you didn't know? Yeah. And remember I showed that picture where there were those little holes? Well, the cystic fibrosis transmembrane protein is not in the nucleus. It's in the cytoplasm on the membrane on the outside of the cells. It's not exactly a hole, but in some respects it's a transporter. It regulates chloride ions. And those things have to be regulated to get a proper balance, let's say osmotic pressure, proper consistency of the aqueous fluids going through your cells. It's an extremely important protein. And there it is. It goes on the outside of the cell to the inside of the cell. And look at this thing here. These little sugars. Remember we talked about sugars that are added after translation. We talked about that in the context of protein C. Talked about that in the context of factor VIII. Well, the CFTR gene, the protein for the cystic fibrosis transmembrane ion channel, has these carbohydrate side chains as well, and I'm just pointing this out, that are virtually critical, not so much for their function, but how they get into the membrane after they're made. So these little sugars that are put on this protein after it's made are sort of like a traffic cop of the cell. They tell the protein where to go. And there's a whole network of membranes in the cell called the endoplasmic reticulum, which bacteria don't have. And in the endoplasmic reticulum is a whole way of getting proteins inside the cell to where they're supposed to go. It's like a subway system. And of course, like a subway system, there has to be specific stops and specific locations. And in some respects, very conceptually, these carbohydrates that are added after translation tell this protein to go to the membrane and stay there because it's very important for getting ions from the outside of the cell to the inside of the cell, from the inside cell to the outside of the cell. Now, there's a lot of mutations of this gene. Remember we said the bigger the gene, the more the chances for mutations. And so here are all the known mutations that have been found by sequencing individuals with cystic fibrosis, that gene. You isolate the gene, much like you did on the take-home test. You sequence it, and you look for differences, and you compare it to the normal gene. And lo and behold, as you'd expect, the mutations are across the entire gene. That's what you'd expect by chance and chance alone. But remember, that poses a big problem for genetic testing, Albert. Because that means that every family is going to have a unique mutation. And so that means that when you're screening for cystic fibrosis, you have to screen a battery of mutations across the entire gene. Does that make sense, Nancy? So you have to screen for a lot of RFLPs or SNPs across the entire gene because the fact of the matter is that any given family is going to have a unique mutation. However, right over here there's a small deletion. And that small deletion occurs in 70% of all of the families that have cystic fibrosis. And we talked about that, and that's called a founder effect. Remember we discussed that? We said that if it occurred way back when, and if it occurred in a particular family, and that family migrated to a particular place and founded the population, it would remain in that population and be a high fraction of the individuals in that population. That's exactly what happened. So cystic fibrosis shows a very strong founder effect. For that particular mutation, which occurred way back when, just stayed in that particular family and that population, and particularly Northern Europeans, and therefore it's a very high percentage. And so the consequences of this are that you have to screen through the whole gene, but you can look for this deletion prominently if you know the background. And that's why people say, what is your origin? Where did you come from? You know, what is your ancestry? Because it helps genetic counselors and people like Michelle Evans who do these things in terms of what kind of screen to give. Chris, grab a microphone and hit me with some questions.
Is it possible to have that one mutation and not have cystic fibrosis?
Is it possible to have that mutation and not have cystic fibrosis?
Yeah.
Only if you're a carrier.
Okay.
If you're heterozygous for that particular allele. If you're homozygous, no, absolutely not. Then you're going to manifest the phenotype.
Okay.
But it's recessive because if you have one copy of a normal allele, it's going to produce a normal cystic fibrosis protein, and it's going to be enough because there's many of them in the membranes to compensate. Tiger, hit me. Faria, do you have any questions? Paying attention all the way up there in Davis?
Yes.
Good. Go ahead.
So if 70% have that specific deletion, is the other 30% all of those little boxes up there? Or could you
And
have
there
that
could be
deletion
others
and
as
then also have...
Yeah. Oh, go ahead.
I
Yeah,
guess-
the other 30% would be everyone else and people from different ancestry as well as Northern Europeans and stuff like that. So the other lines are other individuals of which they have found mutations in these parts of the genes in other individuals and in other families. So those are the 30%.
Do they sometimes have both?
Be very rare, but it's possible. In other words, you could have another mutation occurring after this deletion. It's absolutely possible that you could have 2 mutations, or maybe 3, but it would be extremely rare. Julie.
Are every single one of those recessive, or are there any dominant
All
ones?
recessive. There's no dominant ones because these are called essentially loss of function. If you perturb the protein in any ways, the protein can't function. And therefore, if you have one good protein, it takes over, and therefore the normal situation is dominant to the disease situation, the abnormal. Okay? And it behaves just like everything that we've been discussing in class, alleles and markers and all of these different kinds of things. But again, here's this protein. Each of these parts of the protein, the reason it's displayed like this, correlates with all of these exons. These are the coding regions. This coding region specifies this part of the protein. This coding region specifies that part of the protein. And here are these facts that I don't have to go over. Okay. Now, how do you treat cystic fibrosis with gene therapy? The way you do it is you make a construct. You engineer a good cystic fibrosis gene in some kind of a vector. You do not use naked DNA. Why, Marvin?
Because it's inefficient.
Why? Give Marvin the microphone. I don't know what inefficient means, but I mean that's might be one reason, but what would be the main reason? Inefficiency is a good reason, but what else?
The
Naked DNA.
The human genome might not take up the DNA, and if it does take up the DNA, it might go into...
Think it out critically, Marvin. We've talked about it. Naked DNA, what's going to happen to it? Look how it's being sprayed into someone's nostrils here. What's going to happen to that naked DNA? Has to go down to the lungs, which is where cystic fibrosis really manifests itself by building up lots of mucus because you're not able to regulate the viscosity of the fluids in the lungs and everywhere else. What's going to happen to that naked DNA?
It's just going to get like filtered out and not...
Filtered out.
Or-
What does that mean? What's DNA made of, Marvin?
nucleotides
Okay, how are they bonded together?
with hydrogen bonds. And
The nucleotides are in the DNA molecule by hydrogen bonds. How many weeks have you been in this class? What do you say, Samantha? How are these nucleotides bound together?
Well,
in the DNA?
phosphodiester bonds.
Yeah, phosphodiester bonds. So the pointer is, what do you think, Katie, is going to happen to the DNA if you just use naked DNA in this process?
It's not going to make it.
What does that mean?
It's just gonna die.
Gets lost or something, or?
No, it just has no way of getting there if it doesn't have something to a vessel.
But
to take
what's
it
going to
there.
happen to it?
It'll die. Or
DNA doesn't
alive,
die.
But
It's not alive. It's not breathing. It doesn't have life.
so nothing will happen to it. It would just.
Do you agree with that, Albert? What do you think?
It'll probably get denatured by your body heat.
Could get denatured by the body heat? Maybe. But what might be the most likely thing to happen, Elaine?
Something in your body could cut it up. I don't know.
Yeah, something in your body could cut it up. Now, can you say that more scientifically?
Restriction enzymes.
And we don't have restriction enzymes. You know that. This is the 8th week of this class. You know that eukaryotes don't have restriction enzymes, I hope. If you don't know that, then I've been a massive failure this quarter. So what's going to be degraded, Faria?
Something in the DNA.
Well, what? What will be, yeah, Elaine?
I think bacteria in your body could secrete.
Could do what?
Secrete something.
That could do what? You're being too vague. This is the problem you had with your take-home exam. Agreed, Kelly? This is exactly the problem. What's going to be broken? Ben? You had more precision on your oral exam. What's going to be broken?
Is your body going to break down the sugar molecules?
It could, but what would be the main thing that would be broken in a DNA?
Also, the heat would also denature it
But
too.
what would be the main thing that would be broken? Tom?
The phosphodiester.
Yeah, the phosphodiester bonds. They're going to be the main thing. They're going to be degraded. It's that simple. Eden?
Wouldn't it be easier to break down the hydrogen bonds than the phosphodiester bonds?
Probably not.
Aren't they
Because there's
weaker
all
though?
kinds of enzymes in your cell that do all kinds of nasty things.
Okay.
And remember that DNA has to be broken down in your cell to a certain extent because cells die and they're replaced. And so therefore you have to have enzymes that are going to break down everything in these cells when they die. You have to have enzymes that break down RNA, enzymes that break down DNA, proteins, all kinds of things, so they're used all over again. And so there's things that will break down DNA, but the most likely thing is going to be these phosphodiester bonds. In any case, you want to use a vector. And I'm not going to get into the vector right now that's used for cystic fibrosis in vivo somatic cell gene therapy, but you engineer it into a virus. In many respects, it's adenoviruses, which live in the lungs. These adenoviruses are these particular viruses here. They're naturally found in lungs. You can get them in with aerosol spray. They're, of course, crippled in many respects, so they don't cause what they would generally cause, a cold or something like that. But these adenoviruses are put in there. They go into the lungs. The extra gene, which is the cystic fibrosis gene, is then transcribed and then translated. The virus delivers the gene to the lung cells, and then you get a cystic fibrosis protein, and that protein then goes into the lung membranes, and hopefully it functions normally to start causing a normal regulation of the chloride ions, so that the individuals that have cystic fibrosis now have much reduced symptoms. Now, of course, you're not going to be able to get this into the heart. You're not going to be able to get it into any of the other cells of the body. The most serious manifestation is in the lungs. But keep in mind that cystic fibrosis affects systemically many, many, many different parts of your body, your heart, and other things. And so therefore, it's able to overcome some of the manifestations of cystic fibrosis, but not entirely. But clearly, this is a very, very, very powerful treatment. Again, it's still experimental, but it's done. So that's the first example I'm going to give you of in vivo somatic cell gene therapy. A second example is one which I think is a little bit different, but just as important and just as novel. Brain cancer, like what Senator Edward Kennedy has, is 100% terminal. There's absolutely no cure. very, very, very few people ever survive from brain cancer. In any case, it's a devastating disease. I can tell you from personal experience, not that I want to get morbid on any of us, my father died of brain cancer when he was only 52 years old. It's a miserable thing to see. Dr. Evans just told me that her uncle just died of brain cancer. We had a colleague in our department that died of brain cancer a few years ago, a wonderful developmental biologist. I'm only telling you this because it's a devastating disease. And it's not a fun way to die, because you wind up being paralyzed, you wind up being blind, you lose all of your bodily functions because this tumor just completely takes over your brain, and eventually you succumb. And you can succumb, depending on how it's treated, within 6 months of having it be detected. And so, just as an aside, this is why you really need to read the articles for discussion tomorrow. Because with real good genomics, one might be able to detect these things very, very, very early and hopefully be able to have targets for a particular treatment. Did you guys put in your handout that article that I gave you on sequencing the glioblastoma patients? Good. Most recently, there was an article in Nature earlier this year, in which they sequenced the genomes of 3 individuals, the complete genome of 3 individuals, their glioblastoma tumors while they were still alive. And what they found was, as you will see tomorrow, that there were very specific gene changes in their genomes, different ones for different patients, but they all affected, as I told you last week, the control pathway of cell division. And by doing this sequencing, they could find very specific targets for what would be some very nice therapies that hopefully could perhaps at least treat, not cure, this awful, awful, awful disease. And so these glioblastomas, there's very, very, very little treatment. And you can do surgery, you can do chemo, you can do radiation, but the outlook is very bleak. One of the experimental therapies, and this is kind of esoteric, so you're going to have to bear with me. One of the experimental therapies is to insert in the tumor what would we call a suicide gene. That is, you do in vivo gene therapy by inserting something into the tumor that can only function in the tumor and causes something to kill the tumor. Conceptually, the way it works, Eric, is that you insert, let's say, a gene, a foreign gene, via a vector, which I'll talk about in a second, into the tumor, and this vector only replicates in the tumor. It's a virus. And this virus only replicates in the tumor because the virus only replicates in dividing tissues, and the rest of your brain tissue is not dividing. And so, as you can see in this diagram, you inject it directly into the tumor. This foreign gene that goes into the tumor is slightly different and is completely, let's say, slightly different and completely different from any of the genes in your genome. In fact, it's a gene from a herpes simplex virus, the kind that causes genital herpes and or cold sores. And that has a particular gene in it that's involved in DNA metabolism. And I'm not going to tell you exactly what it does, but when it's in cells, you can treat it with a particular kind of agent called ganciclovir, which happens to simply be a modified nucleotide. And when that herpes simplex viral gene meets up with this ganciclovir chemical, it produces a poison. And that poison kills the cells. And because this gene has not been put in the normal tumor cells, this ganciclovir is completely harmless to normal cells. It doesn't do anything because our cells don't have anything that can convert it into anything which is toxic, because there's no substrate for this particular chemical in any of our normal cells. But in cells that are infected with this gene from the herpes simplex virus, this particular chemical is a substrate for that protein encoded by that gene, produces a toxic substance, and that toxic substance kills the cells. So the way in which this works is you have some kind of probe. Here's the individual that has his brain tumor. You use these retroviruses that I'm going to talk an awful lot about in a few minutes. You've engineered those retroviruses in ways that I'm going to show you with this herpes simplex. It's called thymidine kinase. I'm not going to get into it. Gene. The herpes simplex then delivers the gene to these tumor cells. It replicates in these tumor cells, and when it's replicating in these tumor cells, this thymidine kinase gene is transcribed and translated, and the protein is there. And then what you do is you treat the individual with this ganciclovir intravenously, and the ganciclovir then goes into the brain. The thymidine kinase from the herpes simplex sees the ganciclovir. It's its substrate. It binds to it. Chemical reaction occurs. It changes it. A toxic compound is made, and boom, tumor cell's gone. And it works. Limited, but it works. So it's an experimental therapy to try and target tumor cells and not affect the normal surrounding cells. It's kind of like what we talked about with antibodies. In other words, using an antibody that would be specific for the tumor. In this case, that's how Senator Kennedy was treated. He had an operation, his tumor was taken out, antibodies were engineered to the outside of his tumor, and then he's been treated with his own antibodies to his tumor, and hopefully that shrinks the tumor. And remember I told you last week about Herceptin and how one engineers these antibodies using either fungi or bacteria, cancer antibodies, and they go to the site of the tumor and kill it. Well, this is not an antibody, this is a gene, and this is a suicide gene that when it interacts with the substrate, it produces a toxic substance in a cancer cell specific way and destroys the cancer cell. Highly experimental, but you're willing to try anything with this devastating disease. Someone had their hand up. No? Yeah, go ahead, Eden.
If it doesn't kill,
can you speak up?
If it doesn't kill all of the tumor cells, then what?
It'll come back. It's going to come back. No, all these treatments, it's going to come back. Okay, and it comes back. And that's the problem with these things. So clearly, it's called suicide vector, and you can read that. I'm not going to do it. And that's how it works. And this is where the toxic compound is made conceptually, right in this particular reaction here. And this compound is made, and it's toxic to the cells. The cells die, and that's it. Okay. Those are the only two examples I'm going to give you of in vivo gene therapy. You can go on the web. You can go to the gene therapy sites at NIH. You can find literally hundreds and hundreds and hundreds of trials of using different kinds of genes for in vivo gene therapy. It's experimental. It's very technical. It's very avant-garde. They work in limited ways, but there's no magic cure just yet. Let's talk about the more successful gene therapy, which is the ex vivo gene therapy example. And I'll give you a couple examples of this. Remember, this is now taking cells out of an individual and engineering their cells. Faria, do you have any questions? Did you understand what I said about in vivo gene therapy?
Yeah.
You sure?
I think so.
Did you read your chapter in the book where it talks about this a little bit?
I read it beforehand, but not...
Okay, we'll read it afterwards, and I think it'll make a lot of sense.
Okay.
Let's go to taking cells out. There's this little boy who's no longer alive by the name of David Vetter. He was born with a disease that we've talked about before called severe combined immunodeficiency, or SCID, S-C-I-D. SCIDS is a disease that destroys your B cells and T cells. And remember we talked about B cells and T cells that make antibodies? We specifically talked about B cells. I gave you a slide that showed you how B cells make antibodies. Well, if you have SCIDS, which again is a Mendelian recessive gene, and in the homozygous situation, your B cells and T cells are destroyed. Brian, if you destroy your B cells and T cells, you have no immune system. And so therefore these people generally die from opportunistic infections because they can't fight off anything. David Vetter lived in the bubble for 12 years. Can you imagine that? Had absolutely zero contact with any human being. No touching, hugging, nothing. Pretty awful existence. And then he died at 12 because he did finally succumb to an infection that got in his little sterile bubble. But the point here is that there wasn't really any treatment. There are some treatments, and there certainly wasn't any cure. But what you can see here is that there's now lots of therapies, gene therapies. They've been around a long time. I showed you a slide the first day of this class. I showed you a slide in which I said human beings have been genetically engineered. The first gene therapy experiment was carried out in approximately 1992 or so on some young babies, and it worked, and I'll show you pictures of those individuals now, but it targeted this particular disease. What is the defect in? The defect is in a gene called ADA, adenine deaminase. There it is. It's an enzyme. It's a gene encoding a very specific enzyme. What does this enzyme do? It's involved in nucleotide metabolism. In other words, you have to make nucleotides and you have to degrade nucleotides. Everything is cycled inside your bodies and inside your cells. And this particular enzyme happens to be something that takes adenine, which is this particular molecule here that has this amine group here, and it takes that amine group off and it makes this molecule and then another molecule, and it goes to degrading these molecules into hydrogen, carbon, nitrogen, that's then scaffolds that are then used over again. We salvage everything that we use in the cell. And if you don't have a good ADA gene or enzyme that catalyzes that reaction, what happens is this thing builds up in abnormal ways in specific cell types. Now you might say, well, adenine really can't hurt anything, but if it builds up to toxic levels in a particular cell, because what's there to degrade it and keep it at a normal level is no longer there, you're going to get toxic levels of this particular purine. And when that happens in B cells and T cells, they die. They just can't survive high levels of this particular purine. And when that happens, individuals have SCIDs. And when individuals have SCIDs, Maura, it's bad news. I mean, these kids die. And it's a tragic death, as you can imagine. And so one would like to then treat these individuals in a variety of ways. The goal, get a good, normal adenine deaminase enzyme into their blood cells, so then the blood cells are going to be able to carry out this reaction normally. The toxic levels of the purine is not going to be there. You're going to have normal levels of the purine, and the blood cells are going to survive. And it's a recessive gene because it's a loss of function. The enzyme no longer functions. And if you supply a good enzyme, the reaction can go. And it doesn't matter if the bad enzyme is there. As long as you supply it with a good enzyme, you can overcome this defect. The gene is also pretty big, 32,000 bases. It's on chromosome 20, 12 exons. The messenger RNA is 1,000 bases, obviously, because its exons are spliced together, and it's 323 amino acids. This is the slide I showed you the first day of class, telling you that genetic engineering wasn't just done in plants, in animals, and fungi. It's also in bacteria. It's also done in human beings. And there's this individual that was engineered when she was about 2 years old, and she's now walking around with a gene in her blood cells that she was not born with. That is a genetically engineered human being. She has a normal ADA gene, although she was born homozygous for SCIDs, meaning she had 2 copies of the defective ADA gene, but now she's perfectly normal because her blood cells have been engineered using ex vivo gene therapy with a normal copy of the gene, and it works. So it began a long time ago, and there's the experiment that I'm going to tell you about. Okay. Ex vivo gene therapy, again, just to remind you, you remove the blood cells from the individual or other cells, but typically it's blood cells because you have to do it outside the body and you have to get the cells back in the body. So mainly it's not done with liver cells. It's not done with skin cells. It's not done with brain cells because you have no way of getting them back in the body, having them replicate. It's done mainly with blood cells because you can get them out. You can have bone marrow transplants. You can do all kinds of good things. Now, I should say that an individual with SCIDs, if they have a sibling or a parent, but mainly a sibling, that would have a tissue type match. You all know about organs that reject because their tissues reject each other because they have antibodies that reject. But it's also possible that they can have a sibling like an identical twin or a normal brother and sister that might by chance and chance alone have a match to the tissue type of the SCIDS affected child. If that is the case, they can have a normal bone marrow transplant. They don't need gene therapy. They would just get bone marrow from a match, so to speak, and that match will then take over. Cells will divide and you'll form a normal ADA. Now, that's an interesting proposition because some individual families that have a child with SCIDs decide to have another child, and by using pre-implantation genetic screening, screen the embryos for the particular tissue compatibility genes that might be identical to the sibling that has this disease, then choosing the individual on the basis of a particular genotype so that that individual then can be born when it's implanted into the mother, and when that child is then old enough, the bone marrow cells can be taken out of that child and then given to their sibling so that that sibling can be normal and alive. And that has happened. It's rare, but it does happen. But in that respect, you are selecting for a particular genotype, and in some respects, it's genotype selection. So now it's not random. You're actually selecting for a very specific combination of genes and doing this on a human embryo and essentially having a baby to save a baby. And many people have ethical issues with that. I don't particularly have them, but it does occur, and some in vitro fertility experts will do that. Some won't. It depends upon the individuals. But in that situation, if you can find that match, which the chances are very low, extremely low for reasons that I'm not going to get into, you don't need to have gene therapy. In any case, this is how this works. The virus you have to have a vector in order to do this. You don't use naked DNA because, not Max, but because Marvin is correct, it's very inefficient. You have to transform the cells in order to get the foreign normal ADA gene into the cells. Naked DNA transformation is very inefficient. Remember, we talked about that in the context of making genome libraries. Remember, we said you use a lambda viral vector because it's much more efficient at delivering DNA to the bacterial cell because it's a natural process. And so you want to deliver the gene to the cells using a natural process, and in animals that natural process is a viral, an animal virus. And you use an animal virus to infect, a crippled animal virus, to infect cells in order to get the genes in. And in this case, you use a retrovirus, which I'll get into in a second. And the retroviruses, one of the retroviruses happens to be the human immunodeficiency virus, or HIV, but there's a whole class of retroviruses, some of which are known to cause cancer in human beings. And in fact, these retroviruses, as you may learn in your discussion tomorrow, but you'll certainly learn a little bit later on, these retroviruses carry a very important enzyme for genetic engineering, and that's called reverse transcriptase. And that's how reverse transcriptase was discovered, and I'll get into that in a second. Here are these viruses. These are animal viruses that you can see right over here, and you can see the relationship of the size of those viruses to a typical yeast cell, a bacterial cell, or a typical other kind of bacterial cell, E. coli versus a streptococcus. You can see they're pretty small things, and they're pretty simple creatures. Whether they're alive or not alive is debatable, but generally probably many of these viruses evolved by escapes of DNA from our genomes or the genomes of other animals and just started replicating. And that's how they're able to survive. They replicate to survive. They have no other purpose but just survival. We have a retrovirus. There it is. HIV is an example. This particular virus has a genome made up of RNA. It's a very small genome of a few thousand bases. It's an RNA genome. It's called a plus genome because the RNA genome is the coding strand. It's the sense strand. That RNA can go into a cell and be translated immediately. Okay? So it's called a plus strand RNA virus. But it's a single strand of RNA, and then it has some other things that I'm going to show you in a second. There's many, many, many different kinds of retroviruses. And so here is the vector that's used in ex vivo gene therapy. In the case of SCIDs, it's very efficient in getting DNA into replicating cells. Clearly, it's not good for everything. It was very good in getting it in those replicating brain cells. It's not going to infect them. You have to get them in there by themselves. But it will infect replicating white blood cells. That's what the AIDS virus does. You don't use the AIDS virus for gene therapy, obviously, but certainly it can get into replicating blood cells. And that's why it's a very good vector for ex vivo gene therapy in dealing with blood disorders like of the immune system and a variety of other kinds of things. Julie.
If we ever find a cure for AIDS, does that open up a huge number of possibilities for retrovirus gene therapy? Because then you can treat it? Or is
I'll talk about that in a different context later on. Okay? Your question is correct, and that is that theoretically one should be able to use ex vivo gene therapy to be able to treat AIDS. But there's a real problem with that. The problem is that the ADA situation, you have a blood cell that has a defective gene. And that defective gene can be complemented by getting a good gene in there to work. In the case of AIDS, you have a whole viral genome incorporated into the genome of the cells. And so you have to be able to, and you can't get it out of there. So you're not going to be able to put any good genes in to complement it. The situation's different because instead of putting a good gene in to complement a bad gene, you have to shut down in the AIDS situation bad genes. You have to shut down many of them. And that's what the viral treatments for AIDS do right now. They prevent the AIDS virus from replicating. But I'll show you another way in which AIDS can be treated using gene therapy a little bit later on. Are there any other questions? Samantha.
Just looking on the chart up there, just wondering, what's a pressure treatment? And how can you take tissues outside of the body and treat them?
How about blood cells?
Okay.
And so you use a pressure gun, kind of like a, like a gun that just and gets things into the cell by a differential of pressure.
Okay.
But it's not very efficient. And it's not used very much in many respects. Okay, let's talk about these retroviruses. It's a class of viruses. It has a very specific structure. It has on the outside a membrane, an envelope, made up of lipids and proteins, very much like the membrane in your cells. And this is why it can get into your cells very easily, because it's like a Trojan horse. It actually can get into your cells, here it's coming out, but it can get into your cells by simply merging with your membranes and being transported into your cells, because the cells basically think that it's recognizing something that should be transported into the cells, okay? And so it has this outside membrane, an envelope of lipids. It's called a lipid bilayer, and it's no different than the lipid bilayer that surrounds your cells. I think all of you probably remember that from your 10th grade biology, that all of your cells have a lipid bilayer that encloses the cytoplasm. Well, this lipid bilayer is exactly the same as the lipid bilayer in, let's say, human cells or other animal cells, but it also has some envelope proteins that you can see out here. And these are very important proteins because they dock. They're kind of like docking proteins that recognize the cell that they infect. And in the case of HIV, there's very specific proteins on the outside of the envelope of the HIV virus that will recognize specific proteins on the outside of a blood cell, and it's that recognition between these little docking proteins and the proteins on the blood cell that says, this is a blood cell, I'm supposed to get in, and then it just merges with the membrane and gets in like a Trojan horse. Just completely subverts any kind of immune system that the cell would probably have. And so these retroviruses work like that. So they have an outer membrane, but then they have an inside series of proteins called the capsid. So inside the membrane is a capsid of protein. And so they have capsid proteins. And then inside the capsid is the genome, which is this single-stranded sense strand RNA and reverse transcriptase enzyme. So we have an outside envelope of lipids and some protein, the envelope protein. We have an inside capsid. The capsid then protects the genome, which is RNA. It's a single-stranded RNA, but inside the capsid is also reverse transcriptase. This is the genome in simplistic style. It has this gene called GAG. It has this gene called POL. It has this gene called ENV. ENV are the proteins around the envelope. GAG are the proteins of the capsid. POL is the reverse transcriptase. It's a very simple genome. Very simple. Here's Rous sarcoma virus. This is one of the most famous experiments in cancer biology. This is a virus that was discovered by a guy by the name of Peyton Rous, a Nobel laureate, who discovered that viruses can cause cancer in this particular case. And the reason why this virus causes cancer is because somewhere back in its evolutionary history, it picked up a gene from the chicken genome, a gene that's involved in regulating the cell cycle, and that gene got mutated, and then when the virus infected the chickens, it delivered that gene back into the genome of the chicken, and because it had that haywire cell regulatory gene, that haywire cell regulatory gene caused the chickens to have cancer. And so, and that's also, as I'll show you, the way in which reverse transcriptase was discovered. But they can be cancer viruses in animals and in many creatures. This is HIV. I want it has the exact same structure as this Rous sarcoma virus that I showed you. It has an envelope with the protein around it. There it is. It has a capsid made up of this protein. It has an RNA, single-stranded genome, and it has reverse transcriptase. And here you see that it's merging with the membrane of the cell, and it's getting in much like a Trojan horse. Now, what is unique to the retrovirus? What is unique to the retrovirus, Catherine, is the fact that the minute this genome, this RNA genome, gets into a cell, it's translated, and the proteins, one of which is reverse transcriptase. And that reverse transcriptase immediately makes a cDNA copy of the RNA genome, and then makes a double-stranded DNA copy of the RNA genome, and that double-stranded DNA is then transported into the nucleus and gets integrated into the chromosome at random. And so the cell that's infected with a retrovirus is immortalized with that particular viral genome forever. You never can get rid of it. So people that have HIV have an HIV genome in their white blood cells, and it's there forever. There's a lot of different animal viruses. I don't want you to go away thinking that all animal viruses work like this. You know, polio virus has a double-stranded RNA. Simian virus 40, SV40, that infects monkeys, has a double-stranded DNA. There's all kinds of different animal viruses. Single-stranded DNA, double-stranded DNA, sense-strand RNA, double-stranded RNA, antisense RNA. They're all different classes. But this particular class, the replication is, RNA gets into the cell, it's translated, reverse transcriptase is made, the reverse transcriptase catalyzes the polymerization of a complementary DNA using the RNA as a template, complementary DNA. That DNA is made double-stranded. It goes to the nucleus through those little holes, Tom. It gets integrated with other enzymes into the chromosomes, and it's there forever. When it's in the nucleus, the gene's in now this viral genome, but it's a DNA genome that's part of the genome of the cell that it's in is now transcribed. The viral mRNAs here are translated, and you get the envelope proteins, you get the GAG proteins. They assemble into the capsid, they assemble around the RNA, and then the membranes fuse, and the cell, the virus then is blebbed off. And that process is called getting it in. The process is called endocytosis. You may have heard this term. And getting it out is called exocytosis. These are common cell biology terms. And here you can see the virus blebbing out of this chicken cell here at the end of its replication cycle. Hiromi, did you understand that? Is that clear? So, the point here is that this reverse transcriptase was discovered by David Baltimore, the former president of Caltech, Nobel laureate, along with Howard Tiemann. Independently, they discovered reverse transcriptase in 1975. That was a major discovery, because before that time, Nancy, people thought DNA, RNA, protein. Reverse transcriptase is discovered DNA, RNA, RNA to DNA, RNA to protein. Central dogma of Watson-Crick was modified because enzymes were found that were able to catalyze the polymerization of DNA using an RNA template using reverse transcriptase. Derek, what's been the importance of reverse transcriptase in genetic engineering? What has the discovery of reverse transcriptase allowed us to do? Speak up, speak up, speak up.
It allowed us to make cDNA libraries from mRNA.
Yeah, so it allowed us to clone specific messenger RNAs. I think it's off. It allowed us to clone messenger RNAs. Without the discovery of reverse transcriptase, we'd be in bad shape from a genetic engineering point of view because we wouldn't have been able to clone specific messenger RNAs. We wouldn't have been able to then isolate many of the genes because, as you know, it's easier to clone the message, like the insulin message using cDNAs. Insulin gene using cDNAs. Making insulin in bacteria using cDNAs. Without the discovery of reverse transcriptase, it would have set us back a long time in our quest to understand how genes function at a basic level or with respect to just being able to do anything. It's been a major tool of genetic engineering. Steve.
Looking at the...
I can't hear you because of the microphone. Go ahead.
Looking at the
Thank
previous slide with the cycle for HIV,
yeah.
Does the cell not go through the lytic cycle then?
No, the cell's not destroyed like a bacterial cell. Don't confuse this with a bacterial virus.
Right, I'm trying to clarify.
Yeah, the cell stays alive until, the virus overwhelms the cell and then destroys it, of course. I mean, if you're making tons of viruses inside that cell, it's going to destroy the white blood cell, yes. But it's not lysis in the sense that you learned in a bacteriophage life cycle. There's just so many of the viruses, it just, it kills the cell, essentially. Any other questions? Okay. You with me, Michelle? Okay. So there's the life cycle, and I'm going to go over it a couple more times so you get it. Because it's important for gene therapy, and you'll see why I'm being so redundant on this. Here's the virus. Here's the reverse transcriptase. Here's the envelope. Gets into the cell. The envelope is merging with the membrane of the cell, and then the protein capsid goes in like a little shuttle. The RNA is released from the capsid. You can see that there's reverse transcriptase bound to the RNA. It starts to transcribe the RNA into DNA, so to speak. We're getting a DNA copy. We get a double-stranded DNA copy. It gets integrated randomly into the host chromosomes. That presents very serious gene therapy problems that I'll talk about in a second. Then those genes get transcribed into the capsids, the envelopes, the reverse transcriptase. They get packaged into the capsid, surrounded by the membranes, and they go out of the cell. And that's the life cycle. Derek, you got it? Reverse transcriptase has been studied in incredible depth. This is what it looks like in an X-ray view. This is the structure of reverse transcriptase. This is what it looks like in a conceptualized view. Notice it's like a glove. It's holding its substrate. The substrate is the RNA template of the virus and the deoxyribonucleotides, which are used as a substrate to make the DNA copy. It's very different. It's very similar to what we've talked about before. And you can see here's the RNA, here's the newly synthesized DNA, and I'm not going to talk about exactly how it works, but again, everything is antipolar. The RNA goes 5' to 3'. Reverse transcriptase goes from the 3' to the 5'. It's 5'3", anti-parallel. This is a miniature double helix. Miniature double helix right here. Anti-parallel, as you can see, and all the rules that we've talked about work and operate. Amelia, you happy? Good. Lauren, you happy? Everything clear? Okay. Now let's get complicated. How do we do the gene therapy? And I want you to listen to me. You will have this on your oral exam. There's absolutely no question about this because it integrates everything. So let me ask you a question. Brian, you with me, Faria? Taking a lot of notes?
Yeah.
You're writing away? Your hand is just aching from taking all these notes?
Hurting like crazy.
Yeah, I know. So Brian, if you want to engineer the genome of this virus to have an ADA gene in it, okay? Where do you do all that cloning? Do you do it in a white blood cell? Do you do it in a bacteria? Where would you do all of your genetic engineering manipulation?
In a bacteria?
Good. How would you do that conceptually? Would you be working with the RNA genome or the DNA genome of the virus? What would you be manipulating?
I guess the DNA genome.
You guess?
The DNA genome.
And what would that DNA genome be in if you're working in a bacteria?
In a plasmid.
Exactly. So you've cloned the genome as this double-stranded copy. You can do that because you can isolate the viral RNA, you can use reverse transcriptase, you can make a double-stranded copy, and then you can add C's and G's and do all the things you learned in discussion, okay? So now it's in a plasmid in a bacterial cell, right? Now you want to add the ADA gene. And remember, you have to get it now expressed in the human cell. What would you need to do conceptually?
I would need to insert the gene.
Okay.
But it would have to be within the viral DNA.
So you're going to insert the gene in the viral DNA someplace, okay? And how are you going to get this gene, this ADA gene, to be active in the human blood cell?
I will have to use some kind of on and off switches.
Yeah, and maybe that could be from the virus because the virus does work in the human blood cell, or you could add something else, but probably you just use what's there from the virus, okay? Now, this gene, though, Brian, you're doing really great. This virus has this gag, pull, and envelope gene. You think you'd want to keep that in there when you're doing gene therapy?
No.
Because, you know, if they were there, what would the virus do in the white blood cells?
It's going to produce more viral
Yeah, it would
parts.
start replicating. So what would you want to do before you added the...
I would want to take those out.
You're absolutely right. How would you take them out? What would you need to know?
I would need to know the sequence that...
Exactly. You'd need to know the sequence. And then how would you snip them out?
User restriction enzyme.
Exactly. Fabulous. Absolutely wonderful. Perfect. And then you'd put in its place what?
My gene.
Kind of like what we did with the Lambda virus. Remember, we got rid of some stuff and put some stuff in? Yeah. And so now you have something that may be able to replicate in these cells, insert inside the white blood cells, but it only has a normal copy of an ADA gene and doesn't have anything else. So it's a crippled virus, okay? Excellent. So all that's done inside a bacterial cell. But in order to do the gene therapy, Brian, just to finish this up, do you want to work in bacteria or do you want to work in white blood cells?
In bacteria.
To do the gene
therapy?
therapy?
Gene therapy? No, in white blood cells.
Okay, so where are you going to get the white blood cells from?
Bone marrow transplant.
Transplant?
Oh, not a transplant. Just...
From whom?
From the patient.
Yeah, so you're going to want to take the white blood cells from the patient, And now you're going to treat them with the bacteria?
No.
No. So now you got a problem here. Because you got what you have in a bacteria, and you want to treat cells which are animal cells, human cells, you got to bring them together. And I'm going to show you how that's done. Okay? Excellent. That was that was perfect, actually. So here's how we do it. And here's how it is done and was done. Here again is the life cycle of the retrovirus. There's one thing that I haven't told you about, and this thing is called psi. Right over here, this little red box. This is very, very important. Remember I told you that everything is done by sequences. Well, this little sequence psi tells this viral RNA, when it's made from the DNA genome of the virus in the cell, it tells it to package with those proteins. This is the thing that allows the proteins to wind around the RNA genome. It starts off the whole packaging process. Is that clear, Lauren? Without that psi sequence, these proteins cannot, under any circumstance, bind to the RNA and package with it. Okay? That's a very important fact. So this psi packaging sequence is really critical. So here's how ex vivo gene therapy goes. You do all your cloning, like Brian said, in bacteria. You make a cryptic virus. That is, you disarm the virus by getting rid of all of these GAG, envelope, and pol genes. You replace those genes with your therapeutic gene, so to speak, in this case, the normal copy of the ADA. But the viral genome still has, some of the stuff that's necessary for replicating the virus and integrating it inside the human cell. And you leave in there this packaging sequence. And you do all this cloning in bacteria, which means you're working with a double-stranded DNA. You with me? So that's over in this laboratory over here.